So thank you very much. Thank you for having me. Actually, my first Pichia conference, but I've heard all about it from Rochelle, as you heard last night, and I hear you're a wonderful community, and so far from what I've seen, I'm so happy to be here. So, I guess next slide. I guess I can't change it myself, right? So I think Rochelle's told you about the work we've been doing in cell-free protein synthesis, but in case this is your first meeting as it is mine, I thought I'd start there. So essentially, cell-free protein synthesis is taking the internal contents of the cell and using it to express recombinant proteins in the absence of having to do transformations and grow up cells and induce them and things like that. So it has some uses that we're interested in protein biomanufacturing, in that you can synthesize proteins on demand. You can make these extracts from cells and store them in the freezer and then just take them out when you want them. You can do it anywhere in the world if you like. It's basically a chemical reaction. And this leads to something that's produced very, very rapidly. So for example, if you have an urgent need to make a protein, you can use the system and have the protein in your hands in as little as 3 hours. It has very enhanced controllability compared to a cell-based system. You can add reagents to help your protein fold. You could add non-canonical amino acids if you'd like those to be incorporated and things like that. And if your protein is cytotoxic or challenging to produce, it can actually be easier to get high yields in protein synthesis in cell-free compared to cell-based production. And because it's an open reaction system, you can manipulate it very easily. So for example, we're interested in expressing virus-like particles as vaccine targets, and we can do things to manipulate the reaction environment to promote the self-assembly after the protein has been made. This is a field that has really gone on since the 1960s, undergoing a bit of a resurgence in the era of synthetic biology, and a lot of people are focused on E. coli. And these systems have very, very high yield. In fact, I lost Rochelle to a lab that works on E. coli CFBS just because she can make so much more protein there. But for eukaryotic proteins, it's actually something that we are interested in is, can we make these systems retain their organelles and have post-translational modifications to the protein, for example, glycosylation, which I'll talk about today, but also other things like disulfide bond formation, et cetera. And what this would do is it would allow us to screen proteins in cell-free protein synthesis and retain the key activities. So right now, a lot of the proteins that we are interested in are glycoproteins, but when we make them, they have no glycosylation on them. And this gives us the potential to prototype, decide which variants might be of use for us, and then go ahead and make our cell lines afterward. But I don't want to leave you with the impression that this is a perfect technology. There are definitely challenges. So in general, there are lower yields in cell-based systems apart from the cytotoxic protein example, because when you lyse the cells, all of the components get diluted, and so the reaction rates are slower, and the yields that you get from synthesis are a bit less. And actually, you also lose secretory pathway transport, which means that although you can, in some instances, get attachment of the glycans, you can't get further elaboration from them. So next slide, please. So in case you're not aware of this field, there are basically 3 ingredients to a cell-free protein synthesis reaction: the cell lysate, which is basically the internal glue from inside the cell; the reaction mix that contains the small molecules you need for protein expression, so these are nucleotides to produce the RNA, amino acids, and of course an energy source; and then your DNA template, which encodes for your protein of interest. And there are 2 different kinds of systems that you can run. So you can couple this together so that in the single test tube you get first the production of RNA and then the translation of it. Or you can do it as a linked system where you prepare your RNA separately and then add it just for a translation. And this is becoming, again, something that people are interested in, as we all know now that you can make messenger RNA in high yields, and vaccines can be produced from that. There are also another layer of this, which is that you can run your systems in batch, which is what we tend to do, or you can have some kind of a continuous flow which replenishes energy mixes and things like that in order to drive yields higher. And when selecting the origin for machinery, there are a couple of different things that people have done. So there's something called the pure system, where basically cells are modified to add a his tag to every single important protein to them. So things like the tRNA synthetases, the ribosome and stuff like that are his tagged, and then you can purify those from the cell. Or a lysate-based system, which is what we tend to use, which is where you just basically take all of the proteins that are there that happen to remain active after you've lysed the cell. You can choose organisms with special metabolism if you're interested in them, and of course there are a lot of special things that Pichia can do, so that's something that could be of interest. But in general, yields of prokaryotic systems are greater than those of eukaryotic, and this is something that we're actively trying to work on in the lab. But in my opinion, it's important to prototype the proteins that you're trying to express in a lysate that comes from the organism where you ultimately want to express them. Next slide, please. So what I'm going to talk about today is our vision for the self-free production of glycoproteins, which essentially has three pieces to it. So the first is a high-yield self-free protein synthesis system, in this case from Pichia. Then getting initial glycan attachment with high site occupancy using the native ER-derived microsomes from that, and then modifying that further in vitro using purified enzymes to overcome this, the fact that the secretory pathway transport breaks down once you lyse the cells. Next slide. So I grew up, actually my parents had a used bookstore, and I've read a lot, and I swear to you that I'm not the kind of person who reads the ending of the book before, especially not with mysteries, and one can never do that. But I am, in this case, going to begin at the end with the system that we have for basically generating defined end-linked glycans from a starting glycan of our choice. And that's purely because the middle part is the part where we're still working, and so I feel that the talk works a bit better this way. So what we're trying to do then is to take a protein which has some kind of a glycan attached to it and use a series of mobilized enzymes to modify that glycan in order to get the one that we want. And the enzymes that we've chosen come from a lot of different species, as you'll see, but what we do in all cases is we tether them to a solid support so that we can take them out of the product at the end and reuse the enzyme, because it's really difficult to make these, and so we want to use them over and over again as much as possible. This allows us basically to change the glycan or modify the glycan sequentially, step by step, and this gets around the problem of enzyme promiscuity, where glycosyltransferases will work on different sugar structures at the same time and give you a mixed product at the end. And what we have developed has a modular design that allows us to have nearly homogeneous production of a specific glycan, as I'll show you. And you can imagine this being important for a bio-better kind of scenario where you're able to really tailor a glycan, or for fundamental studies where you want to make proteins with different glycan structures on them and understand what the glycan does in that particular protein. Next slide, please. So I'll tell you about a couple of different examples. So some model pathways, one where we made both single galactosylated structure and using MAN3 and MAN5 as an example of enzyme promiscuity. So that's diagrammed up there on the right-hand side. Hopefully you can see the pointer. It's really difficult for me to tell whether you can or not. But starting from a structure with 5 mannose residues on it, you can add a GNT1 to this. And then the natural pathway that you would get for end-linked glycosylation in eukaryotes would trim some mannose residues off before adding another GNT1 and galactosylating. So what you can do then is you could have this pathway operating, but if you have this inside a cell, if the enzyme galtransferase gets to this structure first, you can galactosylate that and you get a mixed product. And what we were able to show, as you'll see in a minute, is that we can treat this sequentially so that we get either one or the other in near homogeneity. We've also taken this all the way out to a single cell-related structure that I'll show you as well. So in developing this platform, we use a series of steps. We use some free glycans as a starting substrate because we knew that these would diffuse well into the enzymes, but we also produced our protein in Superman 5 so that we could have a nice defined starting structure and look at the ability of this to work on situations where there might be steric hindrance from the protein backbone. And we also use some proteins produced in mammalian cells. Please don't boo me out of the room for showing you a little bit of that, just because it's a nice example of the recycling. Next structure, please. So for the enzyme production, we went with a system where we produced most of the enzymes in E. coli, at least the ones that would fold solubly there, and we did a one-step immobilization and purification using biotinylation of the enzyme through Avitag, and then pulling it out with a streptavidin-coated resin. We sourced the enzymes from different cell types, including tobacco, Drosophila human, and Helicobacter pylori. Again, what we did was screen for enzymes that we could solubly express within E. coli in high yields. And using the Avitag, what we did was we immobilized them to the solid support, either a silica bead or a magnetic-based bead so that we could recycle things. The one place where we had a lot of trouble was getting a soluble expression of MAN2. Those of you who've worked with it probably realize that it's quite large, very, very difficult to express. And so there it was expressed in SF9 cells by a collaborator in Toronto. And we used chemical biotinylation, which is a non-specific method, but again, we were able to mobilize it to the solid support. So for the E. coli production, we included a maltose binding protein tag on the N-terminus to improve the solubility. And we co-expressed a biotin ligase in the cells from a plasmid. And so basically we were able to induce the expression of our enzyme of choice. It was biotinylated inside the cells using the ligase, lyse the cells, and then basically pull out the protein from the cell lysate without having to do any other purification steps. So the AVITAG is biotinylated and it immobilizes to the beads. And again, we use both magnetic beads and silica ones. And so basically what you see here, if you can see them, are a bunch of SCS page gels, basically showing that we were able to produce and immobilize all of the enzymes that were necessary for the pathways that I'm going to show you. We do sometimes have a bit of biotin ligase being also pulled down into the beads. We think that that's because it's attached to the enzyme of choice at the time that we're doing the immobilization. Next slide, please. So here's the example from the mammalian cells, and please don't worry too much. I won't spend too much time on here, but essentially we wanted to make sure that we could reuse the enzymes over and over again without losing activity. So for this, we chose the galactosyltransferase as a case study, and we had a full-length IgG produced in Chinese hamster ovary cells, which the lab also works on, which was not fully galactosylated. So in red bars, it shows the sort of starting material there. And what we showed was that we could basically reduce the amount of G0F and G0 structures by incubating the IgG with the GALT and UDP galactose and allowing the enzyme to work and increase the galactosylation. So you see the structures shifting from lowly galactosylated structures to higher ones over time. As the bars get lighter in color, this means it's a later cycle, and you can see that we do sort of lose a bit of enzyme activity over the 140 hours of use, but we are able to reuse the enzyme over and over again. And actually quite a lot of the loss of activity comes from loss of the actual beads. Even though these magnetic systems are good, they're not completely perfect. Next slide, please. So some examples here, we were able to go to different defined structures, and this is some work on free glycans showing the mass spec of the structure. So we produced, the two enzymes here, the GNT1 and the GALT, galactosyltransferase, and we show that we can take the structures to near homogeneity, about 95%. And if we change the order of the enzymes, if we insert ourselves the MAN2 in between, which again was produced in SF9 cells, you can see that we can make an entirely different structure if we want, again, relatively homogeneous there. And this is on free glycans. On the next slide, we used Superman 5 to produce a model protein to show that this would work when the glycan was actually attached to the backbone of the cell. So here we used a monomeric FC domain, so this is just basically half of the constant domain of an antibody. This expresses very, very well in Pichia in general and in Superman 5 as well. What we were able to do was basically to purify this using a his tag attached to it and send it through basically a set of 4 sequential enzymatic reactions, GNT1, MAN2, GALT, and then sialyltransferase from Helicobacter pylori. And again, you can see here from the Molotov results, we get pretty much near 100% conversion across all steps. Next slide, please. Okay, so that was the ending. We know who the killer is. It's the glycans. Now if we move to the beginning, to the development of the Pichia pastoris cell-free platform, those of you who've been coming to this meeting will have heard Rochelle talk about this before. It's a project she started when she was in my group at Imperial. So remember back in the beginning, I told you that there are 3 ingredients. There's the cell lysate, which comes from the cells that you're interested in expressing your protein in, the reaction mix, which are the small molecules needed to power the reaction, and the DNA template. So shown here in red are some of the things that you need to think about when you're setting up a system for yourself. So on the side of the cell lysate, you have to think about how can I lyse my cells to get a high concentration of protein. The more machinery you have there, the more synthetic potential you have in the reaction. And so we compared homogenization, sonication, and bead beating. All 3 of these work. Before COVID, we had access to a one-shot high-pressure homogenizer, which worked really, really well. During COVID, that was in a different building, and so bizarrely, we couldn't go between buildings anymore, so we started using sonication and bead beating, and both of those also work not quite as well in terms of the overall activity, but they do work. If you don't have access to a homogenizer and you want to try this method, it's still possible. We thought about when to harvest the cells to get active machinery, and as you all know, Pichia grows to really high cell density, but it's important that it's in a very high energetic state there, so we started harvesting between ODs 18 and 20. And we did some strain engineering to improve the extract efficiency, and specifically here we overexpressed a single transcription factor to improve the amount of ribosome content in it. In terms of the DNA template design, the one thing that we had to think about that people working with prokaryotic systems don't do is the fact that RNA capping reactions don't happen very well in these lysates. And so for this, we used an internal ribosome entry site based on the cricket paralysis virus, IRAS, and it seems to work well for protein synthesis initiation. And then in terms of what to put in the reaction mix, so we knew what the ingredients were, we just didn't necessarily know the concentrations, and so we used design of experiments to design a reaction mix that works pretty well. Next slide, please. And so the sum of all of this, which I'm not going to show you in detail, is that we get yields of about 100 to 100 micrograms per mil, depending on what the protein is. Obviously, some proteins are harder to express or larger than others. And we've expressed various reporters, including GFP and luciferase, as well as human serum albumin, VLPs based on hepatitis B virus, and now we're working on HPV L1 protein-based things, and these seem to self-assemble in the reaction as well. Next slide. So now we're at the middle. If we want to be able to connect our cell-free protein synthesis platform with the glycosyltransferase modification platform, what we need to do is to be able to make extracts from Superman 5 and then retain the native ER machinery and get a glycan structure on there in the cell-free reaction. So the first question is, can you make active lysate from Superman 5? And the answer is definitely yes. So this is showing luciferase units in the cell-free reaction compared to the different cell extracts. So here's our base case of the FHO1 strain that we're using. Here are two different Superman 5 extracts, one made by sonication, the other by homogenization, and both of those seem to be more active. I will caveat this here and say that I'm not sure that there's anything specific about the Superman 5 modification. It might rather be that these are have two protease knockouts in them, which might actually preserve the proteins in the extract a bit better than the FHO1, which is parent from X33 and doesn't have the protease knockouts. Next slide, please. So then to connect this then to our sugar targeting platform, what we need is to retain the ER microsomes. So these are vesicles that are derived from the endoplasmic reticulum as the cells are being lysed. So basically the ER gets shredded up and it sort of self-assembles into vesicular structures. And what we wanted to do was have a way of retaining these in the extracts through differential centrifugation. So the PhD student who's been working on this project since Rochelle left looked at differential centrifugation speeds to see what kind of microsome profile she could retain in the lysate, and that's what's showed in the two graphs on the right-hand side there. So each color here represents a different centrifugation speed. The top is basically a DLS trace, which gives you the intensity versus the radius of the microsome particles. And you can see that the speed does affect the size of the vesicles a little bit, but not the overall content. As you can see down here on the bottom, the concentrations of vesicles seem to be very, very similar, regardless of the speed that we use. And on the next slide, what I'll show you is that is a nanoflow cytometry trace that is able to basically quantify the particles that are in the solution and also give us an approximate diameter. So you can see that we've got structures between 60 and 100 nanometers in diameter, and we have quite a large content of microsomal fraction. And what we did do is stain these with a dye called ER tracker to make sure that they are ER vesicles and not fragments of cell membrane and things like that. And it suggests that the majority of the particles are actually microsomes. Next slide, please. Okay, so then we need a model glycoprotein, and Farzana joined the lab during COVID, and so therefore was very interested in all things COVID, as many scientists became in 2020. So she wanted to express the receptor binding domain from the SARS-CoV-2 virus, and the construct that we have actually has 3 N-link glycan sites in it, and retrospectively I should have said to her, this is going to complicate our analysis, but I didn't think of it at the time, so she's gone ahead with it. It's small and relatively tractable apart from the N-link glycosylation sites, we have expressed it in the lab in Pichia whole cells before, so we know it will express, and others have shown that it's a great vaccine target, including someone I think who's speaking later in the conference. So initially, we expressed this without any targeting to the microsome, so no terminal signal peptide, and you can see basically from the Western blot on the side, we're able to express this in different lysates made with different centrifugation speeds. Really no difference between the speed or correlation with the speed and the level of expression. Next slide. So then we put a signal peptide in front of it, and we just use the alpha mating factor as a start. And we basically showed that we could express it still. And if we actually do some centrifugation now at high speeds to pull out the microsomes, we can start to see whether things are being processed properly in the R. So on the right-hand side, it sort of shows you the experimental workflow. We run the cell-free reaction in an Eppendorf tube. We are able to spin that at high speeds, use essentially a detergent, Triton X100, to break open the microsomes, remove the microsomal debris, and have basically the internal contents of microsomes in there. And so these are the different fractions, and you can see that basically after the Triton treatment, we're able to release a protein that looks like it's being processed, which would suggest that some of it is going into the endoplasmic reticulum, and the signal peptide is being cleaved off. There's still a lot of protein that's not being processed, sort of right in here, which is coming out with the pellet after the Triton lysis, and we actually think that potentially that could either be protein that's gone unstuck to the outside of the membrane, or that the KEX2 protease activity is not as sufficient as it needs to be for some of the work that we want to do. Next slide, please. So the big question then is this glycosylated at all? Because in order to connect to sugar target, we need some structure there. We can't magic one up. And I can't really answer that, so maybe, maybe not. Right, so it might be really hard to see, but this is basically a Western blot that shows a PNGase treatment assay on the protein that Farzana is making. So we have, in the absence of PNGase, we have something here. In the presence of PNGase, we have something that's a little bit smaller, but we still have quite a lot here. So either our digestion assay isn't working completely, or this is a little bit hard to interpret because we might have some protein that still has the signal peptide attached to it, which is around the same size, and so it's something that we're still struggling to figure out. So I think we've gone about as far as we can with gel-based shift assays, and what we're doing now is trying to develop a purification protocol that enables us to put this in mass spec and just do glycomics and see whether or not there are structures there and calculate our site occupancy based on that. So the other thing that I want to point out is that if our hypothesis that this is just basically a protein with the N-terminal signal peptide still attached to it, it might mean that our site occupancy is low, so we'll be on to the next challenge of trying to figure out how to get that to be a bit higher. So next slide, please. So with that, I'd like to end. I think I probably have caught us up on time. So this is the vision overall. So we want to be able to make proteins in a cell-free reaction with microsomes, get a glycan attached, and then modify that using our sugar target platform. Next slide. Both of these work, little hearts. Next slide. The middle one is still ongoing, and hopefully next time someone will be here to tell you about that. Next slide, please. So I'd like to thank everyone, especially Rochelle, who started this project off, but everyone who's been working on the cell-free and also on the sugar targeting platform, and also the funders. One more slide. This would be shameless if I didn't have the parenthetical part in the peptides. So we're about to hire and advertise some positions. So if you're looking or know someone who's looking for a postdoc position in Pichia, please tell them to reach out to me. If you're here, please have a chat. And also 2 people in my group have virtual posters. I think they might be the only virtual posters, possibly. Okay. So Farzana has a poster on the work that I was telling you a little bit about with more details on how we're trying to get glycan attachment in microsomes. And then Andre's doing some work in Pichia whole cell processes on vaccine production using rotavirus as a target. So please feel free to check those out and book slots with them. And I'm happy to answer any questions.
Thank you. A few questions. I'm going to shamelessly grab one before.
I was going to give you my mic, so.
Come on in. And so you have 10 to 100 micrograms per mil expression.
Yes.
How much of what volume of Pichia cell culture is required to generate that?
So that is per mil of extracts. So that phrase I was quoting is the mil of the cell-free reaction, right? The cell-free reaction is about half extract and half other stuff. And from about a 250 mil culture, we get about a mil or mil and a half of extract. So it's not too bad. It's obviously far less in terms of productivity than you would have with a whole cell process. But I think in terms of having enough protein to be able to play with, so we need about 50 micrograms for for the amyloid glycomics and stuff like that. So our vision is sort of a platform for screening and figuring out what you want to make.
Can you just briefly comment on where you think the limit would be if you optimize it?
Yeah, so I think the biggest problem is the dilution. So the internal contents of the cell is actually super crowded. So you can add things like PEG to try to increase the molecular crowding, to try to get things to find each other in this cell-free reaction a bit better than they do. But I think that's where the limit is. So that most highly productive prokaryotic systems are a mg or 2 per mil. So we could potentially push to a similar amount, I think, there. But it's about 10-fold less diluted than it is inside a cell.
Hi, does your system do trimming and further modifications that typically happen in the Golgi?
So I think in the cell-free reaction, most of that will be basically non-functional. I think for 2 reasons. So the vesicles are mostly ER derived, so there are probably some pieces of Golgi in there, but we have no way to get anything in there. So there's no transport between things. I don't think the enzymes may or may not retain their functionality, but it's really the getting things between the compartments. So that's why we were thinking of having something that we could start with that we know what it is, which is why we wanted to use the Superman 5, but something then that we could further modify in vitro with additional enzymes.
Translation initiation. I noticed that you are using irises for that.
Yeah.
Did you try to remove iris suppressor from Pichia cells to increase the yield?
No, that is a very good idea. No, so I think we can make capped RNA in vitro and use it, but we wanted to have a coupled system, and so for that we needed an iris. But yeah, it's a good idea.
We're actually working with a suppressor of irises in Pichia, and we have strains which don't have them and overexpress them.
Okay.
So
Yeah. If you would be happy to provide those, it would be something that would be very interesting to try, for sure. This iris doesn't use any initiation factors and seems to be pretty platform independent. So if it is something that works kind of well, I think it would be something that people would be very interested.
Yeah.
Yeah.
Thank you.